This episode is produced with support from the National Centre for Biological Sciences.
On the sands of time, stories unfold, treasures from the past to modern lore. Every leaf, every cell, life secrets they tell. Biolore. Welcome back to Biolore. This is a Nature India podcast series where we unravel fascinating stories from the history of biological sciences in India. I am your host, Shubhra Priyadarshini. Packed into lunchboxes, blended into smoothies, and stocked in nearly every Indian grocery store are bananas. Bananas reign supreme on the superfood list. But did you know that behind every perfectly grown banana, there's a fascinating science at play? A science called plant tissue culture that can speed up plant breeding to develop high-yielding, disease-resistant crops in record time. It may sound like science fiction, but it's reality. Plant tissue culture is a powerful biotechnology technique that has been around for decades, allowing scientists to grow plants in a lab using just a tiny piece of tissue. It could be a leaf, a stem, or even pollen in a controlled environment. This allows scientists to create genetically identical plantlets free from diseases and environmental limitations. Unlike traditional propagation methods that rely on seeds, plant tissue culture can produce millions of identical plants in a fraction of that time. Today we delve into two pioneering plant tissue culture techniques, haploid culture and meristem culture, that are transforming agriculture and forestry. Our journey today will take us from the labs of Delhi University, where Shipra Guha and S.C. Maheshwari stumbled upon shoots growing out of datura anthers while culturing tissue to study cell division. And it will take us to the research at National Chemical Laboratory Pune, where 200-year-old teak trees stand solid testimony to these developments.
Plants are very different from animals because literally any part of the plant can make a new plant. So this is called totipotency. Unfortunately, animals don't have. And this is important for plants because while in animals, our germ cells are pre-designed. They are already there even before we are born. Plants can make germ cells from anywhere.
That's biotechnologist P.V. Shivaprasad joining us from the National Centre for Biological Sciences in Bengaluru. The 1920s essentially set the stage for experimental embryology in plants. Scientists realized that by providing the right nutrients in a sterile environment, they could grow plant tissues without contamination. This idea basically took off in Europe, where researchers optimized artificial media with salts, vitamins, and hormones. In the 1950s, when Indian botanist Panchanan Maheshwari entered the field, key discoveries had already been made, like the role of auxins and cytokinins in growth. But the precise balance of these hormones was still unclear. In his Delhi University lab, Maheshwari, originally a taxonomist, turned his focus to embryo sac development, exploring how female gametophytes formed and how sperm cells fused with egg cells, and this laid the groundwork for breakthroughs in plant development. His work on embryo culture induced parthenogenesis, and haploid production sparked a new era in plant development research. Back then, plant breeders around the world were deep into researching haploids, these plants that only have a single set of unpaired chromosomes.
So in this setup of Delhi University, there was also Professor Johri, who was the student of Panchanan Maheshwari. He and Sipra Mukherjee were really interested in finding whether we can study the pollen grains, what they are made up of, what kind of nuclei fuse. And they ended up keeping this pollen in the media. And it turned out that this plant accidentally could become a new plant. So the outcome was quite surprising. They just kept the pollen and it ended up making a new plant. And this is a haploid plant because there is no fusion with the female egg cell. The advantage was not immediately imminent at that time, but we knew that if anybody can make a haploid plant, and it's easy to then convert into diploid by treating with the colchicine, one can fix anything that is made or seen in nature or created in nature, and there will not be loss of this character in the subsequent generation. So if there is, let's say there is a plant somebody observes making big fruit or better flower, and if we now end up crossing with another plant, this character might get now a little bit, they won't inherit properly. They will not be truly same in the offsprings. But if you take a plant and take its, let's say, pollen and make a haploid, and this plant can now mimic exactly what one has seen in nature or one has created in the lab. So this, without this haploid generation, one needs maybe 10, 12 years to make a proper plant. But with this, one can generate with half the time. Let's say 5 years, one can make a plant which has the right trait, which somebody has created or observed, and put it into the plant and make it stably inherited in the when it is grown in the soil.
In India, Shipra Guha applied the technique to rice breeding at the behest of M.S. Swaminathan, didn't she?
Initially, when Professor Sipra did this work, it was in Datura, and nobody knew what exactly one can do with Datura. Datura is only making very strong chemicals, which now we know we can use them for diseases such as Parkinson. But that time it was not a model which can yield a product, which can yield a new line which people can use. So Professor Swaminathan saw the potential, and also there was enormous work on this line going on in China. So he thought, why not do the same thing with rice, which is obviously our number one crop? And he invited her, and they worked together, and Professor Sipra could generate haploid plants in rice. These are some of the first. One can see how Chinese pursued this because Chinese really respected Professor Supra for this discovery. And over the next maybe decade or so, China started producing these lines, hybrids for the first time, started growing commercially, and these hybrids were behaving way, way better than anything that was grown until then. Without tissue culture, one cannot raise transgenic lines in rice and many other plants.
Since then, researchers have made huge strides in developing anthoculture for all kinds of crops: wheat, rice, potatoes, maize, peppers, you name it. This technique has been applied to a wide range of economically important plants, making a real impact on agriculture. And were there any landmark success stories that stood out or even stand out today in how haploid culture contributed to crop improvement in India?
There are a couple of examples. The best example is Indian mustard to retain the oil content, but you have to reduce this nasty smell. I think this technology was very useful in making plants which can now have less smell but equally or better oil content.
Today, plant tissue culture provides the essential platform for introducing and regenerating genetically edited plants.
Yeah. So currently the most interesting application is to use it to edit the genome. So it has been discovered that one can use a protein called Cas9. This originates from bacteria and it needs a RNA of specific type. And if one can insert this into a plant, there will be change in the digits. That is, ATGC can be changed sometimes to the specific residue or sometimes to any other type of residue. And this means if somebody can manipulate the genome like this, one can make the gene to make more protein or completely remove the gene. Basically, one can tinker with this massive recipe book called genome that we have. The most common way of doing it is through agrobacterium, introduce this protein and then this RNA called guide RNA, and then use tissue culture to make a new plant. Now, what people are trying to do is somehow you reduce the tissue culture step to minimum, or introduce in such a way that you can still make a new plant by maybe picking up specific cells, or the tissue culture itself will be of a different type where you somehow in the growing media itself you introduce this protein, make it to go through the roots with the use of nanoparticles, and make this editing in the genome. So the advantage of these methods is that we will be able to change anything, modify, make better crops, both that can produce more yield or better nutritive capabilities, or sometimes making more of medicinally important compounds. All this is possible, and this is where most of the work is going on right now. And people are trying in different crops in India as well. Many groups are trying to improve these crops with editing.
And can it make agriculture more resilient to diseases and climate extremes?
If anybody can find a disease resistant gene and introduce it to a plant, or if there is a natural variant, a plant somewhere which is already quite capable of dealing with the climate vagaries or disease resistance, one can simply use the anther culture to fix this character and get the offspring out of it. So this is the advantage of using anther culture and similar technologies where one can fix this trait. One can also use this to screen large number of germplasm to see which of the ones is better, then fix that character in the subsequent generation. So that kind of work is again very popular in many countries, especially in countries such as Australia. Climate resilience is the key word, and this kind of research is being done.
After Maheshwari's team developed pollen embryo culture, another big breakthrough that really pushed plant tissue culture research forward in India was micropropagation. This technique involves growing tiny plantlets in a controlled environment before transplanting them into the field. Some of the early pioneers in this field included Narayana Swamy at the Bhabha Atomic Research Center, along with H.Y. Mohan Ram, N.S. Rangaswamy, and K.R. Shivanna at Delhi University. Their work was crucial. It helped speed up plant breeding by cutting down the time between generations and increasing the number of plants that could be grown from a single parent. A business model around plant tissue culture began to take root. Meanwhile, at NCL Pune, under Jagannathan's watchful eyes, another story was unfolding, and it started with Ram and Lakshman, two iconic teak plants. They were over 100 years old, standing tall in the Allapalli forest of Maharashtra's Gadchiroli district. Now, what made them special? They were completely resistant to termites and diseases and had impressive girth and produced top quality wood. But the problem? They were too old to regenerate naturally. And despite multiple attempts, officials from the Maharashtra Forest Development Corporation could not successfully propagate trees like them. That's when they turned to scientists at NCL for help, sometime around 1975-76.
But those people they approached that now tissue culture has come in India, and why don't you try this type of thing rather than haploid culture, and that is having more of the academic value rather than why don't you go for application oriented problem. And when they approached us, we had that time in NCL, we had prepared through our local workshop, NCL workshop. We had aluminum hoops, which used to have perspex sheet on the top. You can see from top, and you can put hands from below. And other times you can close those boxes. It used to be sterilized with rectified spirit and all that. And those boxes, along with the test tubes containing our artificial media, we carried them to the locations. And there in the guest house, all these inoculations were done. But the problem is, of course, climbers are there. They used to get us the meristem tip. Problem is heavy contamination. So we have to work on it and to get the sterile and green culture. And that is how in 1980 it was the first report of matured meristem tip culture of tea.
That was Shweta Kendurkar, former NCL researcher who worked on meristem tip culture for 2 decades. Meristem is the fresh, actively dividing tissue around the tips of roots and shoots, and NCL scientists proved that even mature trees could be regenerated in a lab using these tissues. In fact, this breakthrough made NCL the birthplace of successful mature forestry tissue culture, something that had never been done before on a global scale. And it didn't stop with teak. As the forest department's needs grew, the same technique was applied to other valuable trees like eucalyptus and salvadora. This was the turning point in forestry and conservation, proving that even ancient trees could have a second life through science.
And another problem was rooting of those plants because they are hardwood species. So they don't root easily. So that was also standardized that we tried continuous oxygen treatment, we tried pulse treatment, many things were tried and we get the plant. And this is all. Once we got the plant, they approached us that why don't you go for the upscaling of the technology? Not at a very large scale up to 1000 level or 2000 level. That was also a big number that time. And that is how we entered this micropropagation and scaling up and all that. And we did that part. Then NABARD came up with the proposal that why don't you take other species also like eucalyptus and salvadora, and then some agriculture universities are there.
One of the major success stories in plant tissue culture commercialization in India today is bananas, like we mentioned before. Small labs have replaced the costly foreign models, and businesses in places like Pune are thriving using efficient, locally adapted techniques. Farmers can now grow stronger, healthier banana plants with confidence. But it wasn't always a smooth journey. In fact, the early days of tissue culture in India were full of challenges.
See, initially when tissue culture industries came up in India, mostly they were based on the foreign technology. And they had come up with very big greenhouses and very big special laboratories. But in banana, another problem is that also NCL has worked and helped DBT to come up with the solutions. That in banana, if you go after certain subculture, those plants start showing variation. That is their genetic problem. And most importantly, or the most serious problem was virus.
One of the most devastating was the banana bunchy top virus. If a single infected plant was used as an explant, it could silently multiply across thousands of plants in a lab. When these infected plants were transferred to farms, entire banana crops were wiped out. A major outbreak in the late 1990s near Jalgaon in Maharashtra was a wake up call for the industry.
We had worked on this problem particularly because we were approached by many small scale industries and many farmers also. And DBT also intervened. But now this DBT has come up with national certification system for tissue culture raised plants. There we have come up with the protocols that at every step, disease indexing, virus indexing, and how many subcultures these cultures have gone. See what happens for industries, it's easy to take it to higher subcultures also. But then these plants start showing variation. So that type of certification system is there in place. There are committees who visit, they check all the data which is there, they check the plant, and then labels are provided. Before selling, those labels are put on those boxes and then only they are sold. So basically, banana, it is a success story in India.
But Shweta emphasizes the need for crop diversification in banana farming to avoid the risks associated with monoculture.
But unfortunately, at the moment, people are market driven. So market is for banana, even not for sugarcane also that much. Banana, but it is a success story for banana. But another, what I am observing now, I am out of this market, that they are going for Grand Nine mostly. See, they should go for other varieties also, like Basrai. Other varieties also should be cultured. Otherwise, there will be a trait of monoculture. So future is bright, only it has to be taken up in a different manner.
But to immerse oneself in plant tissue culture is like raising a child.
It requires continuous monitoring.
Yes.
See, like you are bringing up your child, that kind of attention it requires.
She hopes the banana success story can be replicated with teak. Going back to Tadoba was an incredible experience for her, visiting the very spots where she and her colleagues had planted the trees felt like reconnecting with long lost siblings.
See, I tell you, first time after 3, 4 years, we had visited Tadoba and we went to the places where our plants were planted. It was really exciting moment to see our plants, like they are our siblings only. Another thing, now after when the technology was upgraded and these plants were put in different fields, and then farmers used to insist, Madam, please come and see your plants. They are growing beautifully. And when you go to those fields and you see them, you say, oh, thank God, you gave me such a beautiful opportunity to work on these plants and add to the nature. Similarly, in case of Salvadora also, nothing grows except for that plant there, and those seeds are the source of income for them. Conditions are very harsh, 50-degree, and no shade also. 2, 3 times I fainted there. But looking at our plants and all those things, we used to be really motivated that yes, we are doing something for the society. And this is what I feel rather than publishing many papers and all those things. What I feel that our plants are standing high in the field. I don't know now, some of them have must have come for rotation also, 20 years old and all that, 93 and all that. But that is the real moment that you feel, yes, you have contributed something to the nature, to the system also. See, national certification system we have come up with. All those small things which we have worked with are really very exciting. And I am really satisfied with my career. It is my dream that somebody should take up peak technology and make thousands of plants.
And as you can see, Indian scientists have been at the forefront of this movement, playing a major role in solving food and nutrition challenges on a global scale. That brings us to the close of the Biolore series. We hope you enjoyed it as much as we did bringing it to you. Please tune in to the other episodes of Biolore, and I hope to bring you another fascinating series very soon on the Nature India podcast. Till then, this is Shubhra Priyadarshini signing off.
On the sands of time, stories unfold, treasures from the past to modern lore. Every leaf, every cell, life secrets they tell. Biolore.
This episode was brought to you with support from the National Centre for Biological Sciences.